I promise I'm back. Good evening once again. Welcome to today's video where we're going to be looking at what we call synthesis of hydrochloric acid. If you find it helpful at the end of it all, please make sure you like the video, comment down in the comment section, and then subscribe. Let's go ahead and get started. So now, in the previous videos, we have been talking about gastric secretions, and we are looking at what we call the hydrochloric acid as one of the powerful content of the gastric secretion. In today's video, we are now going to be taking more of the time and see exactly how hydrochloric acid gets secreted by one of the most important cells, which is going to be residing in what we call the gastric gland. We are going to be following these kind of the objectives and look at what we call the synthesis of hydrochloric acid. We look at what we call the stimuli and even also what we call the inhibitors for secretion of what we call hydrochloric acid. Let's go ahead and get started. Now, the first thing we need to know is that what are that kind of the cell that is responsible for synthesis of what we call hydrochloric acid. That cell is what we call the parietal cell. Okay, so now this diagram here is showing a single cell that is responsible for synthesis of hydrochloric acid, which we are calling the parietal cell. Now, this kind of the parietal cell is going to be having two kind of the faces. This face here is what we call the apical face. Okay, is what we call the apical, the apical face, and this face which is going to be facing what we call the stomach lumen. Okay, it is going to be facing what we call the stomach lumen. Okay, it is facing what we call the stomach lumen. And then the opposite side, this side here, is what we call the basolateral side. Okay, this side here is what we call the basolateral side. Okay, and this side is going to be facing what we call the blood circulation. Okay, so now when we talk about the basolateral side, we are meaning the face which is facing what we call the blood vessels. And when we talk of the apical face, it is that kind of the surface or that kind of the side which is going to be facing what we call the stomach lumen. Okay, so now having seen that, let's go ahead and see how this cell we are calling the parietal cell, how it makes hydrochloric acid. Okay. Now, like any other cell in the human body, it's going to be containing a lot of different organelles. However much I've not shown them here, but you need to know that it's going to be having the nucleus, going to be having the mitochondria, it's going to be having the rough endoplasmic reticulum, and all those other kind of the organelles that are going to be even in any other kind of the cell of the human body. Okay? So now, the most key important thing with this parietal cell is that it's going to be containing a bigger number of this organelle we are calling the mitochondria. It's going to be having tons and tons of the mitochondria. Now, something which is very beautiful is that we have a specific metabolic process that takes place in what we call the mitochondria, and that process is what we call the cellular respiration. Now, cellular respiration, one of the key byproducts of cellular respiration taking place within the mitochondria is what we call the carbon dioxide. Okay, is what we call the carbon dioxide. Now, inside this cell, we're going to be having a lot of water. So now, if this carbon dioxide coming from this, the byproduct of the cellular respiration for the mitochondria, and even also some carbon dioxide coming from what we call the extracellular fluid, can find itself in what we call the parietal cell. So now, if this carbon dioxide reacts with what we call the water, okay, if it reacts with what we call the water, under the influence of one of the most important kind of the enzyme, you know how this enzyme is called? The enzyme that is going to be speeding up the reaction between carbon dioxide and water, that enzyme is what we call the carbonic anhydrase. Okay, so now under the influence of this enzyme, we are going to be seeing carbon dioxide reacting with water under the influence of carbonic anhydrase, leading to the formation of this kind of the acid which we call carbonic acid. Such another beautiful thing. So now this kind of the carbonic acid is going to be unstable. It's going to be a weak acid. It quickly breaks into two kind of the molecules. It is going to be breaking into two kind of the molecules. The first one is going to be what we call the protons, and then the second one is going to be what we call the bicarbonate ions. Okay, these are the two kind of the molecules that are going to be coming as a result of the splitting of what we call the carbonic acid. Now, what is very beautiful is that these kind of the protons, they have their kind of the special pump that is going to be pushing it through on what we call on the apical surface of the parietal cell into the gastric lumen. You know how this pump is called? This pump here is going to be helping in the pushing out of this kind of the protons into the gastric lumen. Okay, so now we have already gotten what we call the proton in what we call the gastric lumen, and this pump is what we call the proton pump. Okay, this pump here is what we call the proton pump. Okay, super very important. But now, for proper functioning of this pump, we need another cation which has to be entering through this pump for the proper functioning of this proton pump. So we shall see where that kind of cation is going to be coming from to enter in this pump for proper functioning. And this kind of the cation is what we call the potassium. So let's go ahead and see how potassium is going to be coming about to make sure that this kind of the proton pump keeps functioning properly. Okay, now this kind of the bicarbonate which has been formed here, we are going to be seeing that it's going to be leaving the parietal cell into the extracellular fluid. So now it's going to be using this channel. So now this channel is going to be now allowing the out movement of this bicarbonate into the extracellular fluid from where it's going to be entering this blood vessel. Now we are going to be having this kind of the blood vessel which is going to be draining blood from the stomach and take it into what we call the portal vein, then to the liver. Now this blood vessel, it can also be called what we call the gastric vein. Okay, it is going to be draining this kind of the blood which is going to be containing a lot of the bicarbonate. So now if you have the blood which is containing high levels of bicarbonate ions, that blood is going to be more of alkaline, and this kind of the mechanism is what we call the alkaline tide. Okay, is what we call alkaline tide. Okay, so now alkaline tide, we are trying to mean that the blood which is going to be leaving the stomach through what we call the gastric vein is going to be more alkaline than the blood which is going to be coming towards the stomach. It's such another beautiful thing. So now as these bicarbonate ions are exiting, we need some other kind of the anions that are also going to be entering in this channel. So now in the blood, you're going to be having sodium, you're going to be having chloride. So now we're also going to be having a sodium chloride in the extracellular environment of what we call of this tissue. Now what happened is that the chloride ions are also going to be now entering through this channel into what we call the parietal cell. So now this is going to be now the bicarbonate chloride exchanger. Okay, now what happens is that these chloride ions which have entered here, they are going to be having a special kind of the channel where they are going to be exiting out of the parietal cell to enter into what we call the gastric lumen. So now this channel here is what we call the chloride channel. It allows the movement of what we call chloride ions into what we call the gastric lumen. Okay, so now you can see now we are going to be having the chloride ions. We even have what we call the protons inside what we call the stomach lumen. Now what happens is that when these two guys meet, they are going to be creating one of the most important, beautiful kind of the baby. And this baby, which is going to be produced by the union of these these guys, is going to be baptized by the name which you call the hydrochloric acid. Okay, so now when these two guys meet, they are creating a baby we are calling hydrochloric acid. Okay, and of course this hydrochloric acid is going to be doing a lot of metabolic process, going to be doing what we call the chemical digestion, like the denaturation of protein, activation of pepsinogen to pepsin, the killing of the microbes that has come along with the food. It is doing a lot. It is doing a lot and a lot of different roles within the stomach. Okay, it is such another beautiful thing. Okay, so now what happened is that we are going to be having a lot of different protons within this kind of the cell we are calling the parietal cell. Okay, so now if we have a lot of the protons in this kind of this cell, it means this cell, there's a likelihood of it becoming acidic, but that one doesn't happen in the real cells. What happens is that on every single cell of the human body, there is this pump which is always pumping out 3 sodium ions and is bringing in 2 potassium ions. This pump is what we call sodium potassium ATPase pump. It is one of the most common primary active transport that consumes approximately 25% of the ATP generated by the body. Such another beautiful thing. So now this pump here is going to be pushing out 3 sodium ions and then pushing in 2 potassium ions. Okay, meaning that this kind of a pump is going to be creating what we call a concentration gradient. Now, what happens that on the basolateral surface of this parietal cell, we are going to be having this pump that is going to be allowing a passage of some little of what we call the protons to come out into what we call the extracellular fluid in exchange with what we call some of these sodium ions, which are also going to be now entering back. Okay, so now we are going to be now losing the protons out through this channel in exchange with what we call the sodium ion. So now this is going to be now more of the recycling process. Okay, it is such another beautiful thing. So now we have said that if this sodium potassium ATPase pump, it is pushing 2 potassium ions into the cell, but of course we also know that the cell is going to be having high levels of what we call potassium intracellularly. So now if we are pushing again more of these potassium ions, that means we're going to be having a lot of what we call the potassium ions. What happens that some of these potassium ions, they are going to be now leaving this parietal cell through this channel here, and this channel here is what we call the potassium channel. It allows by passive mechanism, it doesn't need ATP. So now it diffuses it passive mechanism to enter into what we call the gastric lumen, of which eventually it will finally come over and then finally enter through this pump. So now here now potassium is entering through what we call the proton pump. So okay, so now that is how the proton pump is now going to be functioning, by allowing out movement of the protons in exchange is what we call the potassium. Okay, now this pump is one of the main target of the drugs, or these drugs which we call the proton pump inhibitors, and even also some other drugs which we call the potassium competitive acid blockers. Okay, so now we have 2 kinds of the drugs which are going to be interfering with the working of this pump. And the first one is what we call the proton pump inhibitors, and even also have what we call the potassium competitive acid blockers. Okay, these drugs also are going to be binding on the potassium binding site and block the potassium from entering the parietal cell. By doing that, this proton pump will not be functioning. In that way, you are stopping the production of hydrochloric acid and you'll be managing what we call the peptic ulcers. Besides that, proton pump inhibitors are super very important in blocking this pump from producing hydrochloric acid. The likes of omeprazole, esomeprazole, pantoprazole, lansoprazole, very many kind of the drugs which are going to be under what we call the proton pump inhibitors. And for the case of the PCABs, we have example the most kind of the common one, what we call the vonoprozan. Okay, what we call the vonoprozan. So now it's going to be under what we call the PCABs. It's such another beautiful thing to know. Okay, now having seen the synthesis of what we call hydrochloric acid, let's go ahead and see what are some of those molecules that can stimulate this parietal cell to produce hydrochloric acid, and what are some of those molecules that can inhibit this parietal cell from producing hydrochloric acid, because it's necessary. Okay, now the first kind of the molecule that can stimulate the parietal cell to produce hydrochloric acid is what we call acetylcholine from what we call the vagus nerve. Okay, so now you're going to be having this kind of the molecule which we are calling acetylcholine. Now this acetylcholine is going to be binding on this receptor which we call the M3 receptors. Okay, muscarinic type 3 receptors, and this kind of the acetylcholine is going to be causing what we call a stimulatory effect to the parietal cell, leading to production of hydrochloric acid. Such another beautiful thing. Okay, then the second kind of the molecule that can stimulate the parietal cell to produce hydrochloric acid is what we call gastrin. Okay, so now gastrin is one of the most powerful stimulator of the parietal cell to produce hydrochloric acid. It is going to be binding on the specific receptor which we call CCK2, and it also stimulates the parietal cell to produce hydrochloric acid. Okay, so now gastrin is also going to be exerting a stimulatory effect. Okay. We also have another kind of a molecule that can stimulate the parietal cell, and this molecule also binds on this receptor, and this molecule is what we call histamine. Histamine. So now histamine is going to be coming from what we call the enterochromaffin-like cell, and it also come and bind on this receptor, and this receptor here is what we call the H2 receptors. It will stimulate the parietal cell to produce hydrochloric acid, and this receptor is going to be one of the targets of some of the drugs which we call H2 receptor antagonists, and they block histamine from binding this receptor. If you block histamine, you will not be stimulating the parietal cell to produce hydrochloric acid. And some of the most important drugs that are going to be blocking this kind of the receptor have what we call the ranitidine and the cimetidine. They block this kind of the receptor, blocking histamine from binding there, of which it will inhibit the production of hydrochloric acid by the parietal cell. It is such another beautiful thing. Okay, so now histamine is also going to be exerting a stimulatory effect to the parietal cell. Okay. Then we also have another kind of a molecule which are also going to be exerting an inhibitory effect to the parietal cell. The first one, which is very important in inhibiting hydrochloric acid synthesis by the parietal cell, is what we call somatostatin. Okay, so now this one here is what we call somatostatin. Now somatostatin is going to be binding on what we call somatostatin receptors on the parietal cell, and this one is going to be inhibiting the parietal cell from producing hydrochloric acid. Okay, and then also have another kind of the molecule that can also inhibit the parietal cell from producing hydrochloric acid. Now this molecule is what we call prostaglandins. Prostaglandins. Okay, primarily prostaglandins E2. Now, this kind of the molecule is going to be binding on the receptor which we call EP3. When it binds there, it will also exert an inhibitory effect to the parietal cell from producing hydrochloric acid. Okay, so now that's why sometimes when you give the NSAIDs, they are going to be reducing the levels of the prostaglandins. And if you reduce the levels of prostaglandins, you are going to be now leaving the parietal cell to produce hydrochloric acid. That's why NSAIDs can predispose someone to what we call peptic ulcers. Besides that, these prostaglandins, they enhance the production of the mucus and even also what we call bicarbonate in the stomach wall. In that way, we shall be protecting the stomach from being digested by what we call hydrochloric acid. So now when you give NSAIDs, they are reducing the levels of prostaglandins, allowing the parietal cell to run crazy to produce hydrochloric acid. Such another beautiful thing. So now we also have another kind of the molecule that can also inhibit the parietal cell from producing hydrochloric acid. This molecule is what we call gastric inhibitory peptide, and this kind of the molecule is going to be produced by what we call the K cells. It can also come over and inhibit the parietal cell from producing what we call hydrochloric acid. Okay, however, we also have some other kind of the molecules that also exerts a minor effect in the production of hydrochloric acid by inhibiting the parietal cell. We have the likes of what we call the secretin. Okay, it can also inhibit the parietal cell from producing hydrochloric acid, and even also what we call cholecystokinin. Those cells from the S cell and I cell, they can also minimally exert an inhibitory effect to the parietal cell from producing hydrochloric acid. Okay, it is such another beautiful thing. Okay, so now that briefly covers the synthesis of hydrochloric acid with some of the stimulators and even inhibitors of the parietal cell from producing hydrochloric acid. If you think that whatever I have talked is making sense, you are benefiting, please hit the like button, comment down in the comment section, and then subscribe. Thank you very much for watching. It has been Tumnad all along from the beginning, and of course, as always, until we meet next time.